Well, thank you very much for a very nice introduction. Thank you. So I will spend half an hour to talk about SARS-CoV-2 in Thailand. But so I heard from Chan that most of you are labs people. Am I right? Yeah. And first of all, thank you very much for the organizer for inviting me here. Now, PR especially, and also Chan and Anthony as well for inviting me to be a speaker here. So I want to spend half an hour to lay out some fundamentals for you guys so that you can understand for the rest of the afternoon. Can you show me your hands if you have been working on SARS-CoV-2? Brilliant. Okay, that's great. That's great. So some of you haven't worked with these viruses yet. So hopefully it will be useful to you guys. So we have been living with this virus for about 4 years already. The world has seen, where is it? Okay, so the world has seen a total of about 700 million cases of COVID-19, which is quite a striking number. The death rate is about 1%. And over the past year, the situation has getting a lot better. And it's worst at about like mid-2022. And lots of death happened in the first 2 years of the pandemic. For Thailand, we have seen about 4.7 million cases, and the death rate is about the same, about 0.7%. And as you can see, we did a really good job in the first year of containing the outbreaks in Thailand. We didn't see that many cases in contrast to the world. The death is pretty much reflected in the number of cases. Okay, so today I'm going to lay out some basic facts or information about SARS-CoV-2. And then I was briefed, gave you some details about sequencing technologies, because I heard that many of you are interested in molecular analysis and also sequencing. Am I right? Yeah? But anyway, so first, some basic facts about SARS-CoV-2, and then followed by SARS-CoV-2 sequence data and sequencing technology. And then I will spend about like 10 minutes talking about some applications of sequence data. Okay, so let's start with some basic facts about SARS-CoV-2. SARS-CoV-2 belongs to the family Coronaviridae. And the coronas come from this word, the sun's corona. So this is an electron micrograph of the virus particle. If you squint your eyes a little bit, you can see there's some resemblance between this picture and that picture. And basically, inside the virions are, the genomic materials, and the yellow border is the membrane, and then the red color indicates the spike proteins. So there's some similarity between this and that, and the scientists decided that this viruses in this family should be called coronaviruses. Okay, and this is a genome organization of the virus. Coronaviruses actually have some of the largest RNA genomes, positive single-stranded RNA genome. The size is about 30 kilobases, which is among the largest, if not the largest. And the genome contains 11 open reading frames. And here at this point, between the first open reading frame and the second open reading frame, there is a programmed -1 ribosomal frame shift signals. This allows the ribosome to frame shift and then continue translating the proteins. That's why the first two of are called OF1A and OF1B. And the first two of have got 16 encode non-structural proteins, 16 of them. And one of the reasons why this RNA virus can have such a huge genome is because of this protein in particular, NSP14, exoribonuclease. It basically the correction modules. Other RNA viruses do not have disability, but coronaviruses do have disability, and that's why it can maintain such a huge RNA genome. And this is the spike protein, which is also the main focus of this talk as well. Okay, and so basically, I believe majority of people here are interested in HIV. Yeah, HIV belongs to the Retroviridae family, but SARS-CoV-2 is over here in Nidovirus order. Nido, interestingly, fun fact, Nido come from the word nidus, which means nested. So all the viruses in this order encode or produce a core 3' terminal subgenomic molecules. So this is one of the characteristics of the viruses in this order. Okay, so SARS-CoV-2 is here, HIV is here, which is quite different. But anyway, so let's learn something more about this particular virus. Okay, so first of all, we might wonder, where did this virus come from, right? So this virus was first discovered in the very late 2019. That's why the disease that it causes is called the coronavirus disease 2019. Maybe like on the 20, very late December. And people have found that this virus is very similar to the bat coronavirus from Yunnan, not from Wuhan. So the origin of the outbreak is clear. Was well, the first reported case of COVID-19 was from Wuhan. But so some surveillance found this bat coronavirus from Yunnan, and it's one of the closest virus to SARS-CoV-2. And further investigation led to the discovery of coronavirus in Malayan pangolins, which are also quite similar to SARS-CoV-2 as well. So people thought that maybe this virus came from the bat, with the pangolin being playing some role in the emergence of SARS-CoV-2. In particular, if you look at the similarity of SARS-CoV-2 against these two viruses, the bat coronavirus, RATG18, and the Malayan pangolin coronavirus, in particular, these two guys, for the maybe majority of the genomes, SARS-CoV-2 are most similar to this bat coronavirus, but in particular at the receptor binding domain on the spike protein, which play a very critical role for the cell entry and host determination. Scientists found that the pangolin coronavirus is more similar to SARS-CoV-2 compared to the bat, which is here, coronavirus. So for example, if you look at the phylogeny or the family trees, are you familiar with family trees or phylogeny? You understand this graph, right? Brilliant. So this is SARS-CoV-2, and if you build the phylogeny from the receptor binding domains, the closest, well, the suggestive closest relative of SARS-CoV-2 would be the pangolin version, and then the bat. But if you look at the synonymous sites only, then the bat still being the closest relative of SARS-CoV-2. So the story is quite confusing. Scientists believe that maybe the bat, maybe the pangolin. And then recently, a study came out looking at, well, investigating or revisit the same question using some more sophisticated analysis tools. And then they suggested that actually the bat lost the receptor binding domains via recombination, but then the ancestor of the SARS-CoV-2 and the bat and the pangolin coronaviruses actually already had this particular version of receptor binding domains. So it's quite a funny story, and I think it is quite interesting. So this is something that you can actually learn from sequence data. So you will see again and again various application of sequence data. Okay, so at the beginning of the outbreak, some scientists suggested that, and also from the past experience with SARS-CoV-2, SARS-CoV-1, and also the MERS, some scientists suggest that actually we don't have to worry about much about this virus because coronavirus is an RNA virus, but with a proofreading ability. Yeah, so these viruses are very well known as a slow mutating RNA virus. If you look at the rate of mutation of this particular virus, it is quite low compared to other RNA virus like HIV. The mutation rate of HIV is about 100 times faster than the rate of mutation of coronavirus. By the way, the rate of mutation, just to be clear, is the error rate per replication cycle. Yeah? And okay, fine. But then actually, the mutation rate and evolutionary rate are not the same thing. Yeah? Evolutionary rate is the rate of change of the population as a whole, and it's not just the function of the error rate of polymerase. So you can imagine that, okay, fine, this virus mutate quite slowly per replication cycle, but if the virus replicates frequently enough, in the population of the virus itself will nearly always contain some mutants so that natural selection can still operate quite efficiently. And based on a recent data on this age, the rate of evolution of SARS-CoV-2 has been estimated to be very much comparable to HIVs. And this is one of the work, some of the work demonstrating the high adaptability of SARS-CoV-2. And this is the work that I did together with Ajahn Aruni, who unfortunately is not here today yet. So at the time, Ajahn Aruni, who also worked with, over there, just enter the room. Well, we work together because we are working in the same department. And she helped a lot with various SARS-CoV-2 and COVID-19 research project in Thailand. And one of the things that she had to do was to grow viruses in her lab. And she found that sometimes it was very difficult to grow SARS-CoV-2 in her lab. So at the time, we wanted to investigate like what happened. And there were various cell lines that can be used for grow SARS-CoV-2 in the lab, including Aiewsakun, tempress 2, and color 3. I will not go into details. But, and then what we did was, okay, let's passage these viruses, just only five passages and see how it goes, yeah? Because sometimes we found that after 5 passages, viruses grew quite well in the lab, but not in the first passage. And we wanted to investigate that. And we found that actually there was some adaptation or selections going on. If you look at various the number of polymorphic sites, the position in genomes with various kind of version of nucleotides, you will see that at the first passage, there were quite a great number of polymorphic sites. But then as time goes by, the number of polymorphic sites decreases, meaning that there was some selections going on. And this phenomenon can be observed across all 3 cell lines. And when you look at the intra-sample variation, this is what you can see. So for example, here, we looked at 2 strains of SARS-CoV-2, and you can see that there was some, the color signifies the frequencies of the mutants compared to the original clinical sample. By passage 4, the population of the viruses has changed already. These graphs show the change in the virus at the what's called an MBCS, multi-basic cleavage site of fluorine cleavage site in viruses that are that were grew in that were grown in Vero E6, I believe. Yeah, and when we found many sites that show some signature of cell line adaptation. So this work at the very least showed that SARS-CoV-2 actually highly plastic and can change very rapidly, in line with the fact that actually these viruses evolve very rapidly, even though they mutate quite slowly. And this ability, rapid evolution, leads to the emergence of various kind of variants of concern. These are some of the past variants of concern. And I'm sure you have heard of alpha variants, gamma variants, beta variants, epsilon, yada, yada, yada. Well, basically, these viruses can mutate quite quickly, and each of them have got its own genomic signature. And some people, well, many people actually have investigated the origin of these variants of concerns. How did they emerge? And the very first variant of concern that was found in the pandemic was the alpha variants. Well, the name itself already suggests that is the first variant of concern. And alpha variants was first detected in the UK. And when they look at the phylogeny of the virus, they spotted this very, very long branch. And they found that actually this branch, the rate of evolution on this particular branch leading to the emergence of alpha variant was very high, about like 2.77 times the background rate of evolution of the virus. And on the root to tip distance regression plot, which is this one, basically the RRT plot plots the collection date against the distance of the virus from the root. Okay, and all you need to know is that the slope tells you the rate of evolution, yeah? And the shift in the vertical rate, the vertical shift of the graph signifies that there was some rapid accumulation of mutations. So this study suggested that, okay, the origin of the alpha variants was, well, in terms of evolution, was the rapid accumulation of many mutations in a very short period of time. And they speculated that that accumulation might have happened in the human chronic infections. Okay, and actually this pattern was saw times and times again. So for example here, this is the alpha variants, yeah, the root to tip regression analysis again, collection date, the distance from the root to the sample, yeah. So you can see, you can see some vertical shift over here, and this branch, basically the slope of the branch, well, no, slope of the line signifies the rate of evolution. This one is steeper, faster rate, basically. And then this particular similar pattern was also observed for Omicron as well. Yeah, so it is now quite clear that the origin of variants of concern are pretty much driven by acceleration of accumulation of mutations. Yeah, but then once it's emerged, the rate dropped down again to the background rate, which is quite interesting. Yeah. Again, this study reported similar results with the alpha, the parental branch high rate, yeah. Delta slightly high rate, gamma high rate, background rate quite low. But once it's emerged, all background rate. Yeah. And so yes, I hope that I have convinced you that these viruses can evolve quite rapidly. But the evolution again is the change in the frequencies of the variants within the populations. Yeah. What do you mean by variant, right? So as you can see, this diagram depicts the diversity, the site-wise diversity across the entire genome of the virus. The higher the bar, the more diversity at that particular site. And this region, you can see lots of bars, yeah? And these regions are basically the spike protein coding region. If you zoom in, you can see that the bars are quite high in the NTD or N-terminal domain of the proteins and also the receptor binding domain as well. One of the, some of the explanation of that diversity could be because those two regions can be accessed by the antibodies in the human. So this is the spike protein with the carbohydrate. You can see that the spike protein are well protected by lots and lots of carbohydrates, right? But this region, this is the receptor binding domain in the ARP conformation, and it is very highly accessible by antibodies. So, and then this again, receptor binding domains, it interacts with the host receptor, allows the cells, allows the virus to enter the cells. If this region gets blocked, the viruses die. Basically, it cannot replicate, it cannot enter the host cells. So the virus have some reason, well, I don't want to say it that way, but like there was some selection pressure imposed upon the virus for it to change at this particular site. And also similarly at the NTD or N-terminal domain as well. Okay, so those are very quick background about SARS-CoV-2, and the next section is about sequencing. So at the moment we have about like 16 million genome sequence of SARS-CoV-2 is the highest number that the world has seen for one particular virus, and people have studied this data and then learned a lot from this. There are various technologies that you can use to sequence a virus. I'm sure many of you have heard about Sanger sequencing already, which is the first generation sequencing technology. Basically, this relies on PCR. The fact that it relies on PCR, meaning that you have to know a little bit about this virus in order to decide the primer. If you don't know anything about this virus, you can't really do, you can't really use this sequence technology. What it is good for, when you know a little bit about this virus, yeah, and and when your sample contains a very low concentration of the genomic material, very easy to analyze because it gives you the faster files, quite cheap and fast per reaction. So if you have like 10 reactions, you might want to use this technology. Okay, when it is bad for, okay, so when you don't know anything about what you want to look for, right? If you have like a sample of, from a clinical sample from an unknown cause, then you don't know what you're looking for, you can't design primer, then you can't use this sequencing technology. Or when your genes of interest are highly diverse, if your primer cannot bind to your samples, you can't sequence the region that you want to look at. When you want to sequence the entire genome, this virus again is about 30 kb, the length, but a Sanger sequencing can only give you about 900 to 1,000 bases. Or when you want to investigate intrasample diversity, you can't really use this sequence technology. And again, the next sequencing technology that you might have already heard of is the second generation sequencing technology, which is the other name for this is a short read sequencing technology. The most famous, the most popular one is the Illumina sequencer. For this one, it only gives you 150 base pair per read. This one is per read. You can think of this as a read as well. So it's about 1,000 bases per read. This one is only about like 150 bases per read, very highly accurate นะ if you want to sequence a lot of sequences, then this is a very good technology for you นะครับ the turnaround time is not that great, it's about 2 weeks นะครับ okay, so with this technology you don't need to design a specific primer นะครับ basically if you have your genomic material, what you can do is to chop the genomic material into smaller bits, maybe 1,000 bases, and then tag ligate adapter to your DNA fragment นะครับ that adapter will allow the sequencer to sequence your DNA fragment นะครับ with this innovation you don't need to design your own primer นะครับ because the sequencer will just read your DNA fragment from the adapter itself นะครับ so with this technology you don't need to know what you are looking for, you can sequence pretty much anything in your samples นะครับ it is also quite good if you want to sequence the entire genome of a bacteria or of a virus นะครับ and also this sequencing technology is unbiased in the sense that it can sequence anything นะครับ so you can use this technology if you want to investigate intrasample diversity. Okay, the downside of this sequencing technology is that it is quite expensive per reaction. So if you want to explore these options in your research endeavor, then you might want to have a large number of samples. Otherwise, it can be very expensive. Also, you would need to have quite a high amount of genetic materials in your samples in order to use this technology. Data analysis can be quite complex because the data size is huge. And because the read is quite short, the data generated from this technology is not very good for large structural variation investigation. And now we have the third generation sequencing technologies already. These two look very different. Yeah, this is very small. I forgot to take the sequencer with me to show you guys, but actually it is quite small. The sequencer can actually fit on your palms. This particular sequencer is called ONT or Oxford Nanopore sequencer. This bridge basically, which is quite huge, is a Pakorn sequencer. But essentially it does the same thing. It can give you long reads. Sometimes third generation sequencing technologies are called long read sequencer. Again, very similar to the second gen sequencer. You don't need to know what you're looking for. You can just like sequence everything in your samples. And some sequencer offer a very high mobility. You can actually move these sequencers around. You don't have to go to the lab. You can just bring your sequencer to your own lab. And it is quite accessible. So it's about what, I don't remember, 50,000 baht maybe. You can be the owner of your own sequencer. And also this sequencer gives you real-time data streaming, so you can see your own reads generated in real time. Another key advantage is that this sequencer can give you quite long reads, much longer than 150 bases. ONT in particular can give you about like, from 10,000 bases to 100,000 bases, so very long. This particular features allows researchers to investigate large structural variations and also haplotypes. The sequencer can also detect base modification, particularly methylation. Yeah, and we all know that genomic information is not the only thing that determines the life of organisms. Methylation or epigenetics also play very vital roles in how organisms develop and grow and interact with the environment. Many studies have shown that, have used this technology to investigate methylation pattern and see very interesting results. The drawback is that, again, high amount of genetic materials are required in order to use this technology. For this one, it gives relatively low throughput. You can't sequence that many samples You can sequence maybe like 1 or 2 bacterial samples if you want to investigate or find the entire genome. Yeah. But for virus, for example, you might be able to multiplex maybe like 10 samples. If you want to look, I know. If you want to look at some particular regions, you might be able to do the 96 multiplex sequencing. With the PacBio, you don't have these problems. It is very highly scalable. And also, in addition to the relatively low throughput, this particular sequencer also have a relatively high base calling errors, despite the fact that many people say that the error rates get much, much better. I will show you the results later on. With PacBio, it solved these high error rates by its own technology. Basically, the adapter has a hairpin loop. So the sequencer can just read that one particular fragment many times, and it self-corrects that particular read. But because of that unique features, the read length can be about like much shorter than this. It's about 5,000 bases to about like 20K. That's about it. Another drawback is that the analysis can be relatively challenging and complex. With the SARS-CoV-2, people have developed some very interesting and very cool sequencing techniques called tile amplification enrichment sequencing. Basically, it's merge the first and the second and the third generation sequencing technologies together, which is quite cool. So once you have the very first genome, right? You know what the genomes look like, then you can start design primers. Yeah. And so scientists are very clever. They decide about like 90 something pairs of primers across that cover the entire genome. And with that primers, you can amplify the entire genome. So you can start from a very low, you can study sample with a very low amount of genetic material to begin with. You don't need to grow them in the cell culture, for example. So samples, low amount of viruses, but you now have 96 primer pairs. Then you can use PCR to amplify bits and pieces of the virus genomes. Yeah. And then you can use second generation sequencer or third generation sequencer to sequence the entire genome for you. And this is the results. Yeah. You can see. But the thing is, as I told you, the primer has to be very good. It has to be spot on in order to amplify your genetic material. So there are some, when this virus can evolve very rapidly, so some region within the genome has changed. Yeah. And you can see that some regions cannot be amplified and therefore cannot be sequenced with this technology. So with this technology, you have to update your primers all the time. And this is the real results from Ajahn Arvini's lab. So this is the, instead of using second generation sequencer, we want to use the third generation sequencer to sequence these particular samples using the tile amplification enrichment techniques. So first generation plus third generation. Yeah. And this is the read mapping. You can see a lot of colors. These colors means that the read itself is different from the consensus sequence. The color, the purple in particular, indicate indel error. So even though, yes, right, the substitution rate of ONT is very good now. Yeah? So the base substitution is very good. The indel error rate is still very bad. So if you use this sequencer, it is very difficult. Well, how can I say this? From my own experience, I think the third generation sequencer alone doesn't help, well, cannot be used to investigate intra-host diversity just yet because of this error rates. So, but hopefully this problem will be fixed soon in the near future. Anyway, So the last part of the talk is about like some application of sequence data. So majority of the work here will be my work that I did with various people in my department. So for example, SARS-CoV-2 in กรุงเทพและปริมณฑล Bangkok and metropolitan region, Thailand, in 2020, 2022. So in the database, 16 million sequences, about 30,000 sequences from Thailand, นะครับผม and this is the distribution of the number of cases over time, yeah, and this is the distribution of the number of sequences over time, นะครับ the correlation doesn't look very good, but if you look at the Bangkok metropolitan region or BMR region, the correlation is there, and the slope here is basically the sequencing rate. If you look at the slope, the sequencing rate of the SARS-CoV-2 in Bangkok metropolitan region is about 0.5%, which met the recommended standard for the recommended benchmark for the genomic surveillance. So great. And from the sequence data, we can determine the distribution of the lineages, the dynamics of the lineages. Yeah. So in the first wave, we see these blue variants, most predominantly 8.6. In the second wave, this is Sanam Muay Boxing Stadium outbreak. This one is the Talat Bar outbreak. Yeah. And it was B.1.36.16 variant, this one. Yeah. And in the third wave, the predominant variant was the alpha variant. And in the fourth wave, the predominant variants were delta variants. But at the time, we just talked about delta. But actually, when you look in more detail, you can see that there were two separate variants of the delta viruses. In the fifth wave, there were basically the wave of various Omicron variants. And if you look at the dynamics of the frequency of the variants over time, I told you about the evolution, right? Evolution is the change in the frequencies of the variants in the virus population. You can see drastic change, and the change happened quite drastically. So this graph alone signifies that there was some very strong local competition among viruses. And this is the phylogeny of the viruses. I want to draw your attention to this column. The blue color basically tells you that the viruses are from Thailand. Light blue, Bangkok. Dark blue, somewhere else in Thailand. Other colors, outside Thailand. And I think okay, cool. So this is the overview of the phylogeny of SARS-CoV-2 in Bangkok with some contextualizing viruses. And from based on the phylogenetic analysis, you can actually estimate the effective population size of the virus. And if you correlate the estimated effective population size of the virus from the sequence data, it correlates quite well with the actual number of cases. So this study supports that 0.5 sequencing rate is actually good enough for tracking virus diversity and burden. The Delta virus have some weird trends going on. Basically, it means that the diversity is relatively low compared to the actual number of cases. So this steeper slope indicates that there might be some strong competitions going on, and there was some particular strain of viruses that can actually spread in the population. And in the fourth wave, that was when Thailand get the vaccination rate of over 85%. Yeah, so we speculated that the vaccination actually helped get rid of some of the variants, and then there were only some viruses that can actually spread. The rate of evolution, again, you can see the vertical shift, suggesting that the emergence of the variants of concern were because of the rapid accumulation of some mutations. And these are, if you zoom in, you can see that in the first 2 years, there were most of the outbreaks were, well, most of the cases were from the BMR. But in the second year, the outbreaks were from all over the country. And in the third year, the fifth wave, everything is everywhere. So the viruses in Thailand are similar to the viruses from outside the country, and that matches with the policy of the government at the time. Okay, and with the sequence data, you can do some GVARs. Again, this is the work that I did with Adanaruni. Basically, we found that there were some particular mutations in the NSP6 that correlates with the asymptomatic infections quite strongly. Yeah, I don't think I have that much time anymore. But anyway, okay, this is the last one already, I think. This is the work that shows that viruses evolve quite rapidly and it alters the immunogenicity, which is the theme of today's workshop. Yeah. So at the time, Thailand was, well, we had coronavax, yeah. And this study basically shows that coronavax can actually induce the antibody production quite well, as good as the natural infections. However, those antibodies were not very good at neutralizing viruses. And I think you will hear more about this story later on. And this doesn't just apply to the coronavac, it applies to all the vaccines, Pfizer, AstraZeneca, for example. Okay, so the takeaway message from here is that the viruses SARS-CoV-2 can evolve rapidly and will continue to evolve and also spread. It will stay with us, that's certain. The wealth of sequence data has significantly enhanced our understanding and knowledge about this virus. This is made possible through the techniques such as tile amplification enrichment technologies, sequencing technologies. And there are various kind of analyses that you can do with sequence data, for example, phylogenetic analysis and also genomic epidemiology. You can do some evolutionary investigation, for example, looking at the origin of the variants of concern. You can do the GVARs to identify some genetic factors that might be correlating with the pathogenicity, for example. Okay, that's it for today. Thank you very much.